Right, thanks everyone for coming to this seminar today. So welcome to another semester of school seminars. Before we get started, since this is the first seminar of the semester, I just want to acknowledge that this talk will be delivered from Noongar land and that the Noongar people remain the cultural and spiritual custodians of their land. So Stuart is going to introduce our guest speaker, and there's also people joining us online. Enjoy the seminar.
Thanks everyone, both in person and online. Type away if you can't hear us for any reason. It's my very great pleasure to introduce Mike Edel. Mike and I, and Guy as well, a few other people go back many years. So Mike was a graduate way back in 2000, PhD?
PhD 2000, yes.
Department of Pathology,
yep.
As it was back then. And Mike's done some very, very interesting work over the years, and this has been reflected in a lot of the fellowships and awards that he's had. Visiting Scholar at Oxford in 2018, the French Australian Fellowship at the Institut de Curie, Paris in 1998, Harvard Medical School, the Dersi Fellowship. What's Dersi?
It's a Catalan travel fellowship. Yeah.
Right. In the Beto Lab in 2004, in 2010, the Ramon Cajal Tenureship.
Yeah.
It really allowed you to focus a lot of your work, I think, around the IPSC technology.
Yeah, kept going on that, yes.
So Mike was at the forefront of a lot of induced pluripotent stem cell technologies, which is sort of like still very much the big thing around the world, I think, in terms of regenerative medicine.
Yes.
And that's where Michael and I have had a couple of publications as well and done some work, which has been very, very useful for both of us, I'm sure. He now teaches embryology and anatomy.
I do.
And
try to anyway.
Yes.
Thanks for Tom.
So today, Michael is going to be talking about his advancing cell reprogramming technology to treat particular types of diseases in regards to genetic instability. So, my very great pleasure, Mike. Thank you.
Thank you, Stuart. Thank you very much for that. It's really nice to be back in anatomy. Thanks for turning up Monday. This special seminar. I know Tuesdays is the longest, so I appreciate that. And today, we're all very lucky to be treated to a talk about cell reprogramming and the question of genomic instability in reference to induced pluripotent stem cells. And I've just taken a tenure position in the autonomous University of Barcelona in the Department of Anatomy and Embryology. And it's great because there's a lot of embryologists where I am now, which is a first, and so hoping to be able to use that experience in this new department to help with induced pluripotent stem cell technology over the next years in my research. But it's been a big look for me to be here I studied anatomy here, finishing honours with Jim Taylor in 1988. Charles Oxnard was then the director of the department, and also helped me with my honours thesis at the time. Since then, I've been working in very hardcore molecular biology. So I'm a molecular biologist anatomist now, after 30 years away from anatomy. I've come back full circle, and now I'm teaching anatomy again. I'm involved in anatomy again, and I'm here visiting Tom and Nick, who are helping me develop a teaching course for growth anatomy, which is quite new to me after a career of research. Let's see if this works. I'm doing this right. So, just jumping straight into it, I want to talk to you today about induced pluripotent stem cells, which are almost identical to human embryonic stem cells, which develop from the inner cell mass of the embryo, very early stages of human embryo development, around week 2. And what Yamanaka did in 2006 and 2007 was extract these 4 genetic factors, OCT4, SOX2, KLF4, and c-Myc, from a little mini library of 22 genes that he selected were involved in the embryonic stage of development of animals. And when he took these genetic factors and overexpressed them in a biopsy from a patient So it could be skin cells, or it could be a hair cell, or it could be cells from the urine, kidney cells that are shredded into the urine. Pretty much any cell from the patient could be expanded and reprogrammed into induced pluripotent stem cells using these 4 genetic factors. And then once you had these cells in your hands, then it really revolutionized regenerative medicine, and also provided a new model for studying human disease that we've never had before. You can imagine if this patient had Alzheimer's, you could then make IPS and then differentiate those induced pluripotent stem cells into any cell of the human body. And so by directed differentiation using specific growth factors or genetic factors, you can make cardiac muscle, neurons, lung and liver. And now you have a wonderful model for studying at patient-specific level at the cellular level that you could never do before. And Yamanaka got the Nobel Prize for this in 2012 with physiology of vaccine. You could also study at the cellular level. You could study at the molecular level if you wanted to do genetic studies. You could also screen drugs with these cells. So, for example, if you were making neurons from a Alzheimer's patient, then you could screen that patient-specific cell line of neurons with drugs to see if you could change the behaviour of those cells, thus perhaps finding a new drug to treat Alzheimer's, or certainly providing a method to screen drugs without going into animals. So doing in vitro analysis, saving animal life. And then eventually the idea, and something that really excites me, is to generate clinical grade cells that you could transplant back into a patient and improve the quality of life for patients suffering from diseases where cell replacement therapy was needed. So people that were losing a certain type of cell, and I'll talk about a couple of diseases that my lab is focusing on at the end of the talk. So this all sounds very romantic and beautiful, and but there are a number of challenges in the field that we're all IPS cell labs around the world are working on, and one of them is to reduce genomic instability of IPS-derived cells. So you can imagine going from a somatic cell all the way back, if you like, to an embryonic stem cell-like cell, which is an IPS cell. That reversal of that process is brutal, and you're asking that somatic cell to really rewind its clock back, and you're doing that by hitting it with a hammer with overexpression of c-Myc and OCT4 and SOX2 and KLF4, and you're asking the cell to become an embryonic stem cell again. There is a problem that these cells do not reprogram correctly, and there's a large amount of genomic instability that could cause disease in these cells. So therefore, you'd never get permission to transplant them, or if you're modeling a disease, you're probably modeling it with a very inaccurate cell line because of the genomic instability. So reducing that genomic instability to increase the accuracy of disease modeling, or also to be able to finally transplant them in humans, is a critical topic that we're all working on. I'll talk to you about that today. I'm also very interested in the idea of the immune response of these cells, because if you take cells from a patient and then reprogram them and make neurons and then transplant them back in the brain, which is something the Japanese are proposing to do, and have clinical permission to do that clinically in Parkinson's disease. So to produce neurons that will produce dopamine, and then these little mini pumps can be transplanted back in the brain, and then produce the dopamine that's missing in Parkinson's disease. This has been given approval in Japan, to my surprise. The question is that are those cells immunogenic? We would think they wouldn't be, because they come from the same patient, but after this brutal process of reprogramming, perhaps there is some change in the molecular profile of those cells that we're about to transplant, and perhaps we need to look a little bit closer at that question. And then another challenge really that a lot of us are focusing on is proper differentiation methods. But how can we recapitulate what the embryo does naturally from 2 weeks to 8 weeks in the formation of organs? There's a lot that we do know, but there's a lot we don't know. And so developing these methods where you can take the iPS cells and add growth factors that we know are important signals during embryonic development, and make a cell like a cardiomyocyte or a neuron or a lung cell. How can we improve those methods so that we can get a physiologically and anatomically correct cell before we start transplanting them in humans or start making conclusions from our modelling, which many papers have been published today on. So the question I was asking myself is how can we make genetically stable, high quality iPS and derive cells from these iPS cells? How can we improve the genomic stability? So addressing this first challenge in the field. So just briefly, what Yamanaka showed in 2006 was that you could take skin fibroblasts from a mouse or from a human, and you expand those skin fibroblasts, and then making viral supernatants of the OCT4, KLF4, SOX2, and c-Myc. You can infect these fibroblasts and then overexpress those genes. And so you're forcing those cells to express these genes, which then forces those cells to reprogram into colonies of iPS cells. And that can be done by virus, which have problems. There's mutagenesis, there's reactivation of transgenes, and it's very, very efficient. Or we can also get these genes into the cells by using virus-free methods. So synthetic mRNA is the method that I've chosen, which we all now know about synthetic mRNA thanks to COVID. We know that synthetic mRNA is what we're using for vaccination now, and for 10 years I've invested a lot of my time looking at synthetic mRNA technology and how we can use this to reprogram cells to iPS, and Warren has published this extensively in 2010 and 14. There's a number of advantages here. First is you're not using a virus, so that's the first thing. I think that's you're not going to get this insertion mutagenesis. You're not going to get reactivation of genes because it's a transient expression. There's no insertion of the RNA into the genome. It's a transient expression, and it's very efficient, very much higher efficiency of formation of iPS cell clones from your starting population. That could be important if you're dealing with a very old skin fibroblast from a patient who might be 70, 80, or 90 years old. Having a higher efficiency means a higher chance of success of getting your iPS cell clones. And you can control the dose, so you can transfect in 100 nanograms of RNA, synthetic RNA, you can transfect 1,000, and it's transient. So this dose control is also better than virus, where you're basically hitting the cell with a big hammer, massive overexpression of the gene, which is very non-physiological. So looking at this method in detail, how can we improve it? So I focused on this c-Myc. We all know c-Myc is an oncogene, and Yamanaka has described c-Myc as being very, very important in the process. If you take that away and just have 3 factors, then very often you can't reprogram the skin fibroblasts into iPS. You need c-Myc. If you look at c-Myc, it really is pleiotropic. It affects proliferation, differentiation, metabolism, translation of genes, which then feeds onto different functions of the cells. And there are many pleiotropic effects. In fact, it's published that 11% of all genes now in our body are bound by c-Myc as a transcription factor affecting the expression of these genes. That's a lot of the genome that's affected by this gene. And we do know that a lot of malignant cancers overexpress this factor. So I thought, how can we remove this to reduce all these problems that probably do lead to the genetic instability or the high levels of genetic instability in the cells? In fact, Yamanaka was thinking the same thing, and he used another form of MYC. So there's c-Myc and there's L-Myc and N-Myc, and he used N-Myc, and which is a transformation to efficient MYC. And if he replaces c-Myc with N-Myc, So he can still make IPS very efficiently, but reduce the cancer threat. The problem is in the detail is that c-Myc from a molecular biology point of view still contains a certain sequence in its genome that is not good for the repair of DNA. So it still promotes genomic instability within a cell at the molecular level. Something that he didn't look at. So I proposed Cyclin D1, and it's a long story how I got to Cyclin D1. But I proposed to replace c-Myc with Cyclin D1. And the first point being is that c-Myc actually works upstream from Cyclin D1. So it's one of the factors, that one of the functions of c-Myc is to accelerate the cell cycle, the proliferation of the cells. This factor, Cyclin D1, is a kinase involved in the cell cycle that's downstream of c-Myc, but it doesn't have all those other effects that c-Myc has. Overexpressing Cyclin D1, you can push the cell cycle, which is essential to attaining a pluripotent state. You need to really push the cell to proliferate faster. You can bypass the first checkpoint within the G1 part of the cell cycle, and this is a second checkpoint here, which is regulated by another kinase. And these two checkpoints are critical in where the cell is looking at the genomic stability of the DNA. These checkpoints are very important in telling the cell, do we have on board after the separation of the chromosomes good quality DNA? And if that question is no, then often these cells arrest at the first checkpoint or a second checkpoint as a fail-safe to ensure that cell does not proliferate any further and carry on to its daughter cells, mutated gene and mutated cells. So Cyclin D1, at this stage, we knew his main role was in pushing the cell cycle, which is important in maintaining the pluripotent state. You need to get the cells really proliferating fast. I published in Genes Development that you could use Cyclin D1 to replace c-Myc in a colony formation assay to maintain the efficiency that c-Myc would achieve. So in principle, D1 could replace c-Myc and reprogram cells. What I didn't know was that Cyclin D1 repairs DNA by homologous recombination. And this is another function, and it's something that we're discovering as scientists every day, that genes that we have that have very well-known functions also play a role in other functions you'd never think about. Cyclin D1 plays a major role in repairing DNA breaks, double-stranded DNA breaks, which leads to genomic instability. This is published in Nature in 2011 and also published in Ocular in 2016, that Cyclin D1 does promote DNA damage repair. So when DNA snaps from stress, there's a break, there's ways to join that DNA back together again. And the best way to do it is homologous recombination. And Cyclin D1 is doing that. It's really pushing these cells to repair DNA. So by homologous recombination, the high affinity method of repairing DNA. So now we have two functions of Cyclin D1 that for me was very interesting in answering this question: can we reduce genomic instability of cells? Not only could we reprogram cells replacing c-Myc with Cyclin D1, which I published in 2010, but also we had this exciting new idea that not only was it pushing the cell cycle, but it was also, Cyclin D1 was also repairing DNA. And so in theory, it should reduce the genomic instability. So that's what we next investigated. Just to show you briefly the model, we had a DNA break here where the DNA is broken. Then you have these machineries, these protein machineries, which is RAD51, BRCA2, that attach to the two broken ends of the DNA. And then they recruit Cyclin D1 through a Cyclin A dependent mechanism and rejoin through homologous recombination, which is very high fidelity. It's very, very accurate. And this is what was published in Nature. So off we went, and we made a lot of IPS cells. And we used, of course, synthetic mRNA methods, and we made it with c-Myc or without c-Myc, replacing it with Cyclin D1. So we started to make IPS cells. If you look here, we see a lovely clone of IPS cells that grow out in colonies, attached to the plate. These came from a patient fibroblast skin cell, and by transfecting the synthetic mRNA factors, we can make these beautiful iPS cell lines. And these are just three of many clones. These are 6 clones, many clones that we made. And they're doing immunohistochemistry for protein expression of the pluripotency factors like OCT4, SOX2, TRA181, SSEA4. These are all proteins involved in the pluripotent state. So you wouldn't see those proteins expressed in a fibroblast, which is a somatic cell. You'd only see these proteins expressed if you've got to the pluripotent state. And also alkaline phosphatase is a marker of pluripotency as well. So at the protein level, for both MYC and Cyclin D1 made IPS, we had lovely protein expression of the pluripotency markers, and also by PCR, RT-PCR, we had the RNA level, we had lovely gene expression upregulation compared to a positive control for all the pluripotency markers. And there wasn't much difference between MYC made IPS, which are in red, or Cyclin D1 made IPS, which are in blue, compared to the positive control in black, which is the commercially made IPS cell line. So we're very successful at making lots of IPS cell clones, either with MYC or without MYC, and replacing the Cyclin D1. And at this stage, they didn't really look that different. The D1 made or the c-Myc made IPS, they look very similar at this point of characterization. So we injected these cells. You trypsinize these cells off the plate, and you can inject them into the testes of mice or into the kidney capsule of mice, which is a very rich environment for these cells to thrive and grow and differentiate. And that's a gold standard method to see if you can generate from these IPS the germ layers. So the IPS will then differentiate endoderm, mesoderm, and ectoderm structures inside the mouse. So this is a test, an in vivo test that these cells are indeed pluripotent. Again, these structures were observed in the teratomas in the mice. There was no difference between D1 or MYC. We looked at the chromosome karyotype for all the MYC made and D made clones, and brief message at the chromosome level, we couldn't see any structural changes looking in depth. We started to differentiate the neural stem cells and make very nice, pure, less than pure neural stem cells. This is an example picture of these neural stem cells derived from either MYC made or D1. And again, there wasn't much difference that we could see in expression level of the gene of these neural stem cell gene markers, indicating that either with D1 or MYC you could make very nice neural stem cells. It didn't matter which method. Same for cardiomyocytes. If you're interested in cardiomyocyte biology, you could make very nice patient-specific models, cell lines of cardiomyocytes, which beat in waves, beautiful waves, beating beating myocytes within the plate that were positive for all the markers of cardiomyocytes, general markers of cardiomyocytes, the same between D1 and MYC cell lines. And then when we looked in more depth at the teratomas that we made from the mice, we started to see some small differences between the two different methods, either making them with MYC or D1. And what we saw was first the size. So if you had a MYC-made IPS cell line and injected that into a mouse, the teratoma that came out was much bigger than the D1 made. And this was already sort of a hint that perhaps the D1 made IPS were less threatening in terms of teratoma size or formation of a neoplasm. And if we measured that over time, indeed in blue, this is the D1 clones, they grow out a lot slower, these teratomas, whereas the MYC-made IPS were growing inside the mice a lot quicker, a lot faster, as you'd imagine with a cancer. And staining for Ki67 or MYC1 is a routine marker in clinical pathology labs for the proliferative state of a tissue section. This is something that you would see in brown indicates that you're positive for Ki67, which means the cell is proliferating. You can see this tissue, this structure within the teratoma is really proliferating. It's a sort of picture if you're a pathologist looking at breast cancer, malignant breast cancer. This is the sort of picture that you would get with very, very high indices or index of proliferation. And if you look at normal resting mammary tissue, you might see a picture out in the microscope, something similar to this, where there's very few cells proliferating in the tissue. And if you quantify that, there really is a significant reduction in the number of proliferating cells. So this was really encouraging. If we got rid of c-Myc and just reprogrammed with D1, we can actually get these cells proliferating in a normal state and not in a sort of cancerous kind of state. We moved on into more depth to cut a very long story short. The certain gene family, where SIRT1 is one of the 7 genes, or a few more genes involved in this certain family of genes. The SIRT1 family is regarded as a prognostic indicator of cell stress. So it's a very nice gene. It does have a lot of functions, but it is regarded as a kind of indicator if the cell is stressed or not stressed. And if we measure that over time, this is the starting skin fibroblasts. This is the very early IPS that are forming. And then with time, passage 3, passage 10, so this is like about 6 weeks of growth. What we see is that the expression of SIRT1 is much higher in the MYC-made, in red, than the D1-made IPS. And so we think this window here, the difference between D1 and MYC-made IPS over time, this reduced window of cell stress is leading to reduced genomic instability because the cell is not so stressed, right? At a genetic level, which is what this gene measures. It measures the stress, the genomic stress on the cell. So that was very encouraging. We looked in more depth, looking at gamma H2AX. This is a histone. 2AX is a part of the histone structure of chromatin. And if we look, this is a measure of the amount of DNA breaks, double-stranded DNA breaks. It's like another indicator of the amount of DNA breaks that are going on. If you look at MYC-made IPS in red, you can always see it significantly less in the D at the 2-week, 4 or 6 week, or even neural stem cells differentiated from the 6-week IPS. So these are differentiated cells. We always saw a significantly reduced amount of this DNA damage marker. So this was really direct evidence supporting the SIRT1 and the other evidence in teratoma. But indeed, we're seeing a lot less DNA double-stranded breaks if you reprogram the Cyclin D1 and get rid of this oncogene c-Myc in the cocktail for reprogramming. And here in the control, this is normal skin fibroblasts. So this is the same fibroblast, but we've irradiated with gamma radiation, where you'd be breaking the DNA up. And you can see that here that as a positive control, we're getting very, very high level of genomic instability measured by DNA breaks. And you can see in early stage IPS made with MYC, it's almost the same as radiating cells with very damaging levels of gamma radiation. So we could reduce that with Cyclin D1. This was really direct evidence saying that D1 was reducing the DNA damage in the cell during the reprogramming process. We kept digging further. We looked at telomere length, and we sent the cells to Maria Blasco, who's the director of Spain's best and biggest cancer research institute in Spain. She is Mrs. Telomere of the world. She is really well expert on telomere length and telomerase biology, and she set up a wonderful technique using fish, the old fish technique, fluorescent in situ hybridization, and for telomere, and then you can measure the length of telomeres within each cell. You can see these red dots is telomere, and we could count those and measure the multiple telomere signals. One of the measures that we looked at, and again, if you have less MTS, it means you have a much healthier, less stressed cell, and that again was reduced either in IPS or neural stem cells derived from the IPS, so differentiated from the IPS. So in conclusion, we published this in Stem Cells in 21. Cyclin D1 reduces DNA damage during reprogramming to iPS cells, we conclude that both iPS and neural stem cells have reduced genomic instability when using Cyclin D1 and removing c-Myc. And we think that synthetic mRNA transfection methods using Cyclin D1 in the cocktail and removing c-Myc should become a standard operating procedure globally for making your iPS cells, because this will lead, we argue, to better quality cells for more accurate disease modelling and possibly a higher chance of getting permission from clinical regulators to transplant these cells in human beings, which is going on right now, and it will continue and grow into the future. So in this last part of the talk, the second half, I'm going to talk about having these cells in our hands to move forward now, I think, in a better capacity to start modelling disease and treating disease. And we think that using synthetic mRNA methods with D1 is really the way forward that iPS cell labs should be going, both in research and in clinical settings. So let's just talk about a couple of diseases that I'm working on, so to sort of improve the quality of life. One of them is ocular surface cornea damage. This can happen from an industrial accident, or it could happen from a car accident, or it can also happen from genetic diseases where the cornea becomes opaque. So those cells are differentiating, becoming fibrotic. And the traditional treatment for this is to remove a bit of the cornea where there is the limbal stem cell niche that's right on the edge of the cornea from one healthy eye, and transplanting that onto the diseased eye. And that healthy limbal tissue has limbal stem cells in it. And what that does is, when you transplant it into the diseased eye, it repopulates the limbal stem cell niche, which sits just around the edge of the cornea, and repopulates those limbal stem cells that are not there. So it's a true cell replacement therapy, and replaces the cornea with proper corneal epithelial cells. And so the person gets their sight back. So this is one method that is being used. I collaborate with a very famous doctor in Catalonia, Barcelona. His name is Rafael Badaquer, and his grandfather, or his great-grandfather, he transplanted limbal tissue from the healthy eye into a diseased eye of a Spanish princess in the 20th century, and it worked. So the family became instantly very well connected in Spain, and they built a wonderful hospital in Barcelona and an ophthalmology centre for optic diseases, which is brilliant. What this family is doing for ocular disease is phenomenal. And I've been very lucky to be working with Rafael Badaquer and his team to develop iPS cell technology. So instead of taking a bit of healthy tissue, we can actually take some skin cells from this patient, make iPS, differentiate the limbal stem cells, put them on a fibrin patch on the eye. The cells migrate into the eye and regenerate the cornea. This is the idea. And Dr. Badaquer is very interested in this technology because a lot of the time this method doesn't always work. They don't like to damage the healthy eye. Take a piece of tissue, it's not a good idea. And other methods like artificial cornea leads to inflammation in the long term. So just to talk very briefly, we developed this differentiation protocol. We derived ectoderm or neural tissue from the iPS using a special medium called E6 with factors that are important in the normal development, during the development of the cornea or the ectoderm. And then using another medium containing factors important for the formation of the corneal stem cells, which later
Mike, if you can hear this, you're muted.
This is an example. Is that better? Can you hear me?
Yes.
Okay, great. Thanks for that message. So this is what other people are doing. This is what Takahashi's doing. Pit Coffee in the London Eye Project in England is also doing. We can take IPS, make RPEs and bioengineer. These are into clinical trials and already in humans. So IPS are already in humans. I lost my words here. People are also moving on to the organoid field, and instead of making a single sheet of cells, they're recognizing that the retina is an organ of multi-cell layers, and so they're developing what is still at the research level organoids and tissues, and these are floating in the cell culture medium, and they can structurally get very, very nice retinal structures, organoids look like the retina with the different neural layers and rods and cones. But this is yet to go into any kind of clinical application, but it is something to keep note of. That organoids is a developing field. My lab, another disease we're very interested in is idiopathic pulmonary fibrosis, and that's where the lung becomes very, very fibrotic. To the best of my knowledge, there's not much treatment for fibrosis of the lung. It is a really prevalent and problematic disease that usually leads to a diagnosis, a pretty poor diagnosis, prognosis. And I think current treatment at the moment are steroids, and just wait and see. What I thought we could do was transplant in these fibrotic lungs a lung stem cell, so a tissue-specific lung stem cell that we know is very important in producing protein that is important in the structure of a healthy lung, and very important in the structure of the alveoli of the lung. So again, thanks to embryology, we can take patient IPS, we can make those IPS using the methods I've described, and then differentiate using specific factors in the medium. We've got them listed here at different stages of over time, to definitive endoderm, to anterior foregut endoderm, same stage, the embryonic development of anterior foregut endoderm, where the lung develops from, and then produce, using these factors, FGF10, etc., lung progenitors, and then stop by adding this inhibitor, this growth factor, at the lung type 2 epithelial stem cell. And we get very morphologically very nice cultures of lung stem cells, or type 2 epithelial lung cells. And here all we're showing is that when we do this differentiation process, the pluripotent marker OCT4 is completely switched off in these cultures of cells that are lung stem cells. And we did a lot of characterization, and they are pretty nice lung stem cells. And just to cut a long story short, we took another rat model where they're treated with bleomycin. So bleomycin is injected into the lung that causes lung damage, causes fibrosis and inflammation. That's regarded as a very blunt model of idiopathic fibrosis or fibrotic lung condition. Could be also akin slightly to COPD as well. What we see is this very large fibrotic response. You can see here the destruction of the alveolar buds and the formation of the fibrotic tissue within the lung. This is a reversible process in the rat. After 4 to 6 weeks, the rats do recover naturally. So this model, you have to be very careful when you assess it to make sure that you're looking at a real change and not a natural change in recovery that the rat makes from the chemical damage. And in this photo here, what I want to show you is that when we transplant, we treat the rat with bleomycin, but we transplant with the epithelial, the lung epithelial type 2 lung stem cells, that we can really reduce the amount of fibrosis. We weren't convinced that we were getting the alveolar structure to form again, but we were convinced in seeing quite a reduction in the amount of fibrosis, and that in itself is quite a landmark in terms of lung medicine and treatment. Just to look in a bit more depth, this is the bleomycin treated lung, and we looked at a marker called alpha smooth muscle actin, which is a marker of a fibrotic fibroblast. And if you look at that, you can see that these lungs from the rat that have been treated with bleomycin have a very strong fibrotic fibroblast response. So these are really fibrotic lungs. But when we transplant the lung type 2 lung stem cells made from IPS in these bleomycin treated mice, we can really almost completely remove the formation of these, or reverse the formation of these fibroblasts. These fibrotic fibroblasts. So that's just more evidence. And by Western blot, we also see a reduction to normal at the protein level. So that's just 2 diseases. With Oxford, we're working on a beautiful model of from the same patient, a Parkinson's patient, we're making IPS and then differentiating to sympathetic neurons via the neural crest cell line differentiation lineage, and then also cardiomyocytes, and then co-culturing the two and looking at the synapse of the sympathetic neurons on the cardiomyocytes, which until only the last 10 years was thought never to exist, but we do now know some of the Chinese have actually counted the number of sympathetic neuron synapses on cardiac muscle in animals. Then we now know that synapse plays a large role in arrhythmias, and so we're modelling at Oxford using IPS and using my method of IPS formation arrhythmias in vitro, and this is good because it's helping us understand better drugs that interfere with that pathway, that synapse that's gone wrong in terms of the signaling pathway, and also because it reduces the number of animals needed in research. And traditionally in physiology, animal model is the model used quite widely. And with IPS, we can reduce the amount of screening experiments done by first screening in IPS. But I'll talk about that another day. Just to finish up the last 5 minutes, I really love this question. It's, we do assume there to be no immune response from these cells eventually when we do transplant these in patients. And this is funded by the Ministry of Innovation in Spain. And this is a really great question, I think. If you look what everyone else has been doing, some people say in 2011, with the group of Zal, that there is an immune rejection. So you take IPS and you derive cells, you transplant that back into the same mouse. So it's a synergenic transplantation, right? You get immune rejection. Then Lyle said there is no immune rejection in 2013, and then Araki and we both agreed there was an immune rejection, so that's not good, right? And then more recently, and quite a large study, Morizan in non-human primates transplanted IPS-derived neural stem cells and found a minimal immune response, but not too serious. And Mandai, this is the Takahashi clinical trial, taking the RPE cells, the retinal cells derived from IPS and transplanting them in patients. So this is actually in humans. One patient, they found no immune rejection in the Cyclin trial, and this is like the only information that we have of what's going to happen in humans when we transplant these cells from the immune response. One patient. The problem with all these studies, in my view, although they're brilliantly done, especially the Morizani, is a fantastic piece of science, is that they didn't wait long enough. We know that the immune system is made up of 2 parts, the innate and adaptive. The innate's a little bit longer, and should be really waiting a year or 2. And if you talk to Professor Badakir, certainly for the Cornea, you should be waiting up to 5 years with assessing the immune response. So these studies anywhere between 2 weeks and 3 months only follow-up. So just going through some pretty heavy molecular biology. The model was patient class. We made IPS. to everyone's body, still differentiated to fibroblasts again, and we have what I call was F1 and F2. So F1 and F2 should be the same. The hypothesis here is that these cells, these fibroblasts that come from the patient, should be identical to these fibroblasts, the F2, that were differentiated from the IPS that were made from that patient. So F1 should equal F2. They should be the same in terms of their immune response. And we did global proteomic analysis between these two lines. With this hypothesis, they should be the same. And wherever you see yellow, it means that they're not the same. And so within cell transduction function of the cell, there are large differences between F1 and F2, but also quite some differences in the immune system proteins as well. And if you map it out, what we found with toll-like receptor 3 and 4 was really, among other proteins, this is very interesting to me because these are immune proteins. And I'm not an immunologist, although I'd love to be. It's a really fascinating field, and I've been working on this for a while now. Toll-like receptors are essential for life. If you knock them out, we die. And they've been evolving for millions and millions of years. You can find them in fungi and plants. They're everywhere. For millions of years, toll-like receptors are essential for life, many types of plants and animals. So we did some more screening, this time at an epigenetic level, and we compared fibroblasts of the F1 to the IPS, either made by Cyclin D or by c-Myc, so 6 clones, and then also neural stem cells, Cyclin D made IPS or c-Myc. Basically, the take-home message is that epigenetically, in terms of methylation status, they're very, very similar. Actually, the cells are reprogrammed at the epigenetic state very, very well. But what we did find was this TLR3, again, and this time it was a short isoform of TLR3 that is not properly reset. It's actually hypomethylated both in the IPS and the neural stem cells. There is some return of methylation, but it doesn't really come back in all the clones, especially this particular sequence here. So that's very, very hardcore molecular biology. And I think I'm just going to skip forward on this and just say the take-home message is we need to screen our iPS cell clones because this TLR3 is actually being overexpressed in our cell lines. And TLR3, for those immunologists amongst us, we know that particular toll-like receptor is essential for switching on IL-6, which is the inflammation cytokine. And so what happens is when you transplant these cells, you'll be getting physiological levels of IL-6 being produced by those cells. And you don't want that in the human body. You don't want IL-6 because then you'll create an inflammation status in your body. And as we know, inflammation, constant inflammation is a sign of one of the causes of Parkinson's disease. Right? We don't want a constant inflammation state in our body. So we certainly don't want to transplant any of these iPS cell clones. And for your modeling in vitro, if you're modeling the disease, you have to measure the IL-6 in the supernatant because you might have very high levels of IL-6 growing being produced in your cell lines, and that could affect your model, whatever disease you're studying. Having very high levels of IL-6 could be regulating all sorts of genes in your disease model, making it inaccurate. So I'll just skip that and just finish. This is what we think is happening in a lot of iPS cell lines. This is the wild type TLR3. This is the normal signaling pathway from the cell membrane. It activates IL-6, it creates a pro-inflammation state in the body. And what we found is this TLR3 is overexpressed, but also a isoform. So both the wild type and isoform were overexpressed in many iPS cell clones. And it's not reset epigenetically correctly during the reprogramming process. And that's what we dug out of all this molecular biology at the end. And this isoform is working as a dominant negative and can cause unresponsiveness and anti-inflammation, prevent the anti-inflammation response. So it's a bit complex story, but basically we need to be screening the cell lines for these factors. I'll stop there. I think that's enough. I'm not sure what the time is, but we're pretty much on time. I think we've all had enough of IPS this morning. Thank you for your attention, and I'd like to thank everyone from Spain that have collaborated. And there's a lot of great people here, Maria here, Maria Blasco, particularly Victoria Moreno, and also my team, and the funding, mostly from Spanish Ministry of Innovation history of health. Thank you.
Good timing, Mike. We actually do have teaching in here in about 5 minutes time, I think. So we have some time for some questions. Anybody in the face-to-face audience, Silvana?
Oh, Michael, thanks. That's really interesting. You talked about the innate and the adaptive arm. TLR3, more of the innate
Yes,
early response.
that's right.
And I just wondered whether or not you'd considered some of the more adaptive responses and even those immune cells that may be in the tissue of the experiments you do.
Yeah, that's a great question. So, you know, we only just looked at MHC1 and MHC2 expression by RT-PCR. We didn't really see any differences in expression levels of that major player. So that was about it. So what we were relying on was this objective screening to tell us what was different between patient cells and iPS-derived cells from those patient cells. So we're relying on the screening, the proteomic screening and the methylome arrays to tell us what those differences were. And none of the adaptive, we didn't see many differences in the adaptive proteins particularly, or in the methylome. So, and yet the screen didn't pick it up. Doesn't mean that there aren't any. Just the screen, those omics that we didn't pick it up.
Good talk. Do you use any type of cell tracker to track your iPS cells in especially the fibrosis, the long fibrosis, to see what lineage you actually differentiate your stem cells into? Or better alveoli versus how they reduce the fibrotic cells and at the same time to track the territorial information.
Yeah, so it's a great question. It's sort of leading into two questions. You know, one is in the field of lung stem cell biology, and one is a true lung stem cell. There are intermediate states between type 1 and type 2 that have been characterized by single cell RNA sequencing. But generally, it's thought that the type 2 lung stem cells are the main progenitors that live in the alveolar structure that regenerate and produce the cell surfactant 3, a type C protein that's essential for alveolar structure. So if we had a cell tracker, we could look at that sort of lineage of differentiation, but that's been published. So we're pretty happy that the type 2 is what we should be making, and the delta P53 characterization studies, we're pretty happy we're getting very nice cultures of type 2. The other part of that question, which is a great question, is about, is it a true cell replacement therapy, right? Or is it a paracrine effect? And this is a big question in the field. And tracking your cells once you transplant them, as Jill will know from the spinal cord injury stuff, is that, you want to know where the cells are, if they're still there after a month or 2 months. And so they are a true cell replacement. They actually are going in there and replacing and in the structure of the cell. We did that and could not find them. We looked at, we did single cell suspension of lung tissues. So we made single cell suspension after transplant, and we had the cells labeled with a GFP cell tracker, and we put it through a flow cytometry machine that took a photograph of every single cell, right? And you could see every single cell, and also gave you all the flow cytometry data. And we couldn't see a single green cell in the lungs after transplantation after 2 weeks.
Was it a human cell in the animal model?
Yeah, so we couldn't find a human.
Can you do like human mitochondria fish?
Yeah, you could do that, but we also did PCR, RT-PCR, we couldn't find GFP. So we didn't think we had any cells there after a while. What we're thinking is that was a paracrine effect. So while the cells were, they were injected by a catheter into the lung that physically went in, we think that it must have been a paracrine effect, and then the cells died. So we're moving on with that, and what we think now is better for the lung fibrosis is not a single cell suspension, but a lung organoid, because the lung organoid will probably solve the other problem that we're not seeing, which is the return of alveolar structure, the histological structure. We put cell suspension in there, we get a nice reduction in fibrotic fibroblasts. That's probably a paracrine effect, but we're not getting the alveolar structure to regenerate and make new lung structures. So this is clinically what we really want to achieve. And we think the lung organ is going to be the answer to that.
In the interests of time, I think we better stop to let teaching continue, but in the meantime, Michael, thanks for a really interesting talk.
Thank you very much, everyone.
And thanks very much for attending today, everyone.